My name's Anna Rubio. I'm a PhD student at the University of Bristol, and I'm going to talk a little bit about my work in developing a low-cost device to control microsystems in space and time. So I've been building a device which I call the DOME, which stands for Dynamic Optical Microenvironment. The aims with this device was really to create something that could be used for closed-loop control over microsystems in a similar way to a robotic controller would be for a robotic system. We wanted something that could enact this closed-loop control with high spatial temporal resolution, so really being able to localize to individual agents within the system, so kind of engineer behavior on a local level, but also throughout the system. It was also important that this could be reproduced at low cost in a kind of a DIY fashion to really widen the accessibility of the tools to lots of different areas. So to give you an idea of some of the different things that I could be talking about when I say micro-robot or microsystem, we can think about these examples. So for example, a bacterial cell. A bacterial cell is a really great example of thinking about these kind of agents as micro-robots, because bacterial cells are really tiny computers. They're kind of full of this genetic circuitry that we can actually go in and engineer, which is what people do in synthetic biology, for example, going in and engineering different behaviors within these cells. We can also think about inorganic systems, so like micro or nanoparticles. Obviously, we can't engineer them in the same way that we do with bacteria, but we can do surface functionalization and sort of chemical engineering in a way that allows the particles to sort of gain extra capabilities in a way and interact with each other and with their environment. We can also think about unicellular organisms, for example, algae, and also mammalian cells. So you can think of like epithelial cells or even cancer cells. All of these things could be our microsystem that we're interacting with. Now, the reason that it's so interesting to think about cooperative microsystems is because generally microsystems are cooperative. You rarely get like one cell, one bacteria, one particle. And these things are so small, they come in large numbers generally. And often when they are in those large numbers, there's interesting behavior that can emerge or be engineered. So an example that we see in nature is biofilm formation. So seeing a large number of bacteria come together and form this very tough film, very difficult to kill with antibiotics or to clean. So obviously that's an example that's bad for us, but we can also think about examples that we could want to engineer that would be helpful to us. For example, drug delivery by nanoparticles, or thinking about sort of shaping, morphogenesis, self-assembly of these systems, a whole range of stuff. So that's kind of the motivation behind why we want to control these systems and what we want to control. But how do we go about controlling them? Obviously, you can't necessarily build a remote control or a computer to do it in the same way that you might with a big robot. But there's a few different options. The way that I've gone about it is to think about light as a control mechanism. I think light as a control mechanism has benefits over some of the other methods, which I'll go into. So for example, magnetic control is widely used, or like chemical induction, thermal activation. I think light has advantages over these because of the really high resolution you can achieve. Obviously, for some of them, it's a bit more difficult. And even if you can create a delivery system where, in theory, the resolution is high, with things like thermal or chemical, you're going to get diffusion across your system. So even if it starts out really high resolution, it's not really going to stay that way. It's also very easy to monitor what's going on. If you're using light as a control mechanism, you can just image it. Normally, you can just see it. You can see it in a microscope or in a camera. It's also very dynamic, so obviously it's very quick and it's easily switchable. If you suddenly want to turn a signal off, it's just going to be gone, unlike, again, with like chemical or with thermal. And it's very powerful at small scales, so we can think about things like the photoelectric effect, or the fact that you can make and break bonds using light when you get down to these really small scales. So in thinking about how we start to engineer closed-loop control with light as a control mechanism, what we want to do is shape the light environment, observe the changes, and then analyze those changes for, say, position or certain types of movement, the density of the agents, and use that as feedback in our control loop. So to that end, the device that I've built, you can see here, again, it's called the DOME, so the Dynamic Optical Microenvironment. It uses the same technology as you'd find in an overhead projector. So you'll have a digital light processor here in the form of a microprojector. You can project that through a condenser lens, so you get this small structured pattern on the sample stage in RGB wavelengths. That's then imaged through this imaging column, which contains an objective, a tube lens, and some filtering optics. It's projected onto a camera, which is connected to a Raspberry Pi, which does all of the image analysis. So all the image analysis is done on board and in real time. Obviously, that's critical for closed-loop control. That communicates over a local network with a second Raspberry Pi, which is used to drive the projector. So that's how we kind of get this loop. And you also have some LEDs for backlights and for fluorescence imaging. So in terms of the specifics, the projector operates using a digital micromirror device, which is what you can see here. It's like this little pixel grid. It's kind of almost like a dynamic photo mask. The resolution is 854 by 480 pixels, and you get a projection area of around 14 by 8 millimeters squared. A single pixels measures out at about 20 microns squared, and as I said before, you have these RGB wavelengths. So you have these 3 distinct peaks when we look at the spectral analysis of the light source. I've currently got it configured to do imaging at 2 different magnifications, although this is possible to change. That's 9 times and 90 times, which gives you a field of view of 7 millimeters or 700 micrometers, respectively. All the custom parts, which are kind of all the parts here that you see in black, they're all 3D printed in just standard PLA on a fairly basic 3D printer, so it should be really easy to reproduce. And all the optics, electronics, and computing components, they're all quite simple. They're all quite low cost. So I mentioned obviously I'm using the Raspberry Pis, and again that's with the idea that this should be modular and customizable for anyone who wants to use it. Most of the scripts, all the scripts in fact, are coded in Python on the Raspberry Pi, and the total cost comes in at about £500. So I mentioned the 2 different magnifications. So just to give you an idea, these are both cropped from the field of view. So this is not the whole field of view. This is just a subsection, so you can just see what's going on. If we see here, we've got an imaging volvox here, so a type of algae that are 300 to 500 microns large, depending. So this is the 9 times magnification you can see on the left, and on the right we've got 90 times magnification. So you can see the difference, and you can see that we can start to see some of the nice internal structures. You can also do fluorescence imaging, so there's an example of that here, just imaging some fluorescent bacterial colonies. To actually calibrate the system, so obviously we have the area that we're projecting, and then we have what the camera can see, and we really need to calibrate between that so that we can respond to things that are going on in the camera frame of view with corresponding projections. So if we want to localize light input, say light up one area, we need to know which projector pixels to light up in order to make that happen, basically. So what we want to do is create a mapping between the projector space and the camera space. So if this is a projector space, how we calibrate it is to project 4 points like you see here, and then you get a readout from the camera image. So you get these 4 points read out. And what that allows you to do is to calculate the matrix parameters that you need to transform, to create a transformation matrix between the 2 points. So you get a translation, a rotation, and a stretch. So once you've performed that calibration, then you can run your algorithms. And for example, if you have an agent that's localized here, then you can run those coordinates through these transformations, and then you would get some projector pixels output at the end. So for example, that might be like here. So then we can light that up. So to show you an example of that, here's just an example of communication propagation through some static particles. So this is kind of augmenting the reality of the particles. So what you have is it isolates one particle here in the camera frame of view, runs through the transformation and gives it its own little halo. And then that propagates through. So it then checks, okay, is anything else within that? Is anything else within that? And then it gets to the end and it stops because there's no other particles within that field of view. So that's a static example of what we can do. To show you some dynamic stuff, here we have Here we have this is just a water mite that I found in a local pond. This is just swimming around the camera frame view again, and I wanted to kind of see if it would interact with light in an interesting way. It seemed like it had some attraction to blue light, so I projected this circle, and then what you get is you can kind of see at certain points it seems like it's kind of following around the edge, but you can almost then actually take those frames and kind of analyze them and try and figure out, actually, is that what it's doing? So it's a good way of testing if things are responsive to light, and if so, how, and how do they behave. Another example here is with paramecium. So just these little single-celled organisms. Here I'm just giving them these kind of, again, augmented pheromone trails. So kind of thinking about the way that ants lay trails to communicate with each other and to signal with each other. We're sort of trying to replicate that, but with these cells in kind of an augmented way. So you can imagine if you then have a system that's highly responsive to light, if you have cells that are highly responsive to light, if you start laying these augmented trails, you're going to get a situation where they're either kind of attracted to or avoiding the trails of the others. So you can start to get some really interesting kind of swarm collective behavior out of that. Another nice example I've got here is kind of a replication I did of Conway's Game of Life, but a kind of closed loop version that incorporates real world elements. So the rules of the Game of Life, if you haven't heard of it before, basically any live cell with 2 or 3 live neighbors survives to the next generation. Any dead cells with 3 live neighbors becomes a live cell in the next generation. All other live cells die, and all dead cells stay dead. So what that looks like in simulation is this. So you kind of get these patterns spreading across, and in areas of overpopulation they die, underpopulation they die, so it's only very specific places that they sort of stay alive. So that's what the simulation might look like. And then this is the closed loop version. So you can see up in that right corner, I'm basically seeding it using these Volvox cells. So because there were three within close proximity to each other, we enacted this rule 2. So we started to get patterns forming there, and then that spreads out there. You can see another instance of that that sprung up here. So it's a whole separate kind of colony of life, I guess. So that's a good example of how we can use it to kind of overlay these systems. So it's almost semi-augmented reality, but then also actually being fed by the system and feeding back into the system, where we have agents that do respond to light. So the last thing I'd like to touch on is just some of the thoughts we've had around the idea of taking some of the capabilities of the dome currently and adapting that for looking into cancer research, and particularly thinking about studying tumour environments and maybe being able to study what happens within a tumour environment when certain cells are killed, or certain cells are activated in particular ways that are not homogeneous throughout the tumour environment. So if we have here our tumour environment, then we can think about, for example, using it for direct targeting. So we could optogenetically upregulate or downregulate particular genes, or target the cells directly, so shining light onto the cells in a way that either damages them or upregulates something that changes their properties, but doesn't necessarily affect the cells around them directly. Then we can also think about indirect targeting. So rather than us targeting the cells, we could imagine, for example, here we've got nanoparticles that are distributed throughout the tumour environment. We could imagine those being photoresponsive, so that when we then shine light onto those areas, localized light onto those areas, only the nanoparticles that are within that light would be activated. So you can imagine they then release a drug, or they heat up, or whatever it is that they do. And in that way, we'd still have the localized tumour control, but sort of indirectly through another medium, through these particles. Or you could also think about targeting the extracellular matrix. So we could think about targeting that and sort of getting a secondary response from the tumour environment. So those are just some initial ideas. Obviously, we haven't really gone down that road yet, but it's something I'm definitely very interested in. So if you have any ideas or any suggestions of ways that we could bring that to fruition, then please do contact me. I've got my Twitter handle down here, and you can also send an email, and this is my email here, so you can send it to my Bristol University email. But thank you very much for listening. I hope you enjoyed the talk, and if you have any other questions or anything like that, please do contact me.